Thanks to Benchling for inviting me to talk. Yes, at Editas, we do a variety of CRISPR-based medicines. We do eye, lung, blood, muscle, liver, and oncology pipelines. One of the key eye programs we're working on right now is for Leber congenital amaurosis, which is LCA10's most common factor. And for that, some of the symptoms happen at the first year of a child's life, and it eventually leads to blindness. And there's no known therapies. And so what we're working towards is helping resolve and fix that disease. One of the things I use Benchling for mainly is as a platform. It's really enabled us to build out and extend what we need to do while allowing some of the basic infrastructure to be handled and managed by the Benchling system. Basically, I like to think of it as we get to do the interesting problems, and Benchling does all the basic stuff. And some of the main goals that when we work on stuff, we work on managing the samples. We want to integrate instruments, automate workflows, generate reproducible and reliable data and results, the traceability of the samples and containers, tracking things, improving the efficiency of the scientists, in the lab and also at their desk, right? We want to speed things up and make sure that all the data is centralized. So when we do any reporting or do any data analysis, we're going to one place of the truth, and we're not trying to go to multiple silos and trying to reconcile what all that data is and which is actually the correct. So at Editas, we basically want to do is leverage like a bunch of off-the-shelf tools. Benchling is basically the framework for that. We basically doing cloud first. We use AWS, and we use some Atlassian tools on top of that to help manage and wrap some of the loose ends amongst all this functionality. We basically use almost every kind of function that Benchling uses, from like the ELN, the molecular biology suite, the registry, the assays, the warehouse. Likewise, we use a lot of the AWS stuff. One of my first case studies I'm going to talk about is a sequencing pipeline, and like probably many companies here, do some sequencing, and the way I think of sequencing makes you think of a Anna Karenina quote. Is like, all happy families are alike, but all unhappy families are unhappy in different ways, and sequencing pipelines are sort of like that. Everyone does a sequence pipeline. They're all different. So I'll describe what we do, and yours may be slightly different, but you probably have somewhat similar process. Our old process, you started with a scientist making a sample sheet, which gets fed into the sequencer. It gets processed. It takes 3 days for that to get processed. Then the scientist makes analysis sheets, then lets a computational biologist know that the analysis sheet is ready for them to process. The computational biologist runs his analysis, and then he pushes the data up to S3. And each user-driven step, adds delay. People are on vacation, things happen and finish over the weekend. What could really be simplified is by automating stuff and coming down from 6 to 7 days, we're reduced the process down to approximately 4 days through elimination of all those user-driven steps. And Benchling was a key factor in providing that infrastructure. Today, the user creates their sample sheet, it goes through the sequencer, and then the rest of it just kind of gets pushed through in an automated fashion, using some AWS functions that are heavily leveraging the Benchling Web Services API, and we can just go and call and pull all the information that we need, and they have such a great breadth of services that we can make sure that any piece of data that we need for that analysis, we can pull and get and generate that, and then push it through AWS Batch, and then just push those results right into S3. Another solution or case study that we have is being able to trace all the reagents and samples and containers that we use. And for the sequencing, we end up having to generate these primer plates. And before we built out the solution, the scientists would have to look at the samples and then identify which primers they need for those samples, and then they'd have to go and look to see like what containers or how much in each vial of that primer do they have, and where are those primers, are they even available? And then if they do have enough primer, okay, how many reactions they need to do in order to quantify the total number of primers if you repeat the sample and need multiple primers in different wells. And so it's like a lot of very manual driven steps in order to fully calculate and get that information. And it's very user error prone because people are kind of hand looking and managing. And then if you go and think you have something in the freezer and it's not actually there, then it's a problem. But by leveraging the Benchling web service API, we're able to fully automate that process. And so we just built a simple little web UI where the users will just enter in their sample plates, and then we'll go and query Benchling and look, based on those samples, well, what primers do we have? And we can optimize. We can see how much reaction we need, and so you might even see multiple containers with that primer. But we can make sure we pick the right container that has enough volume, so they only need to pull one tube and not multiple. And we then also generate a pick list we have a Hamilton SAM freezer, and so that can go and go to the Hamilton SAM to pull all those tubes from the freezer automatically, and it'll generate the plates in Benchling for us. So it'll now have those primer plates with the samples and all the primers on it, and we can also report back with any low volume. So if we have some primers now now we've pulled enough off of and we're low on it, we can report back to the scientists okay, we've made these plates, but you're not going to be able to do this again because there's not enough volume, so you might want to reorder and get some more of this primer. And so now not only does it fully automate it, and so by push of a button, it gets a lot more automation and a lot more information back to the scientists, so they can really ensure that they have everything they need in order to continue with their experiment and go ahead with more experiments in the future. There's also some other features in there for, sometimes you want to override what you might have defined, and so there's other features in there, but basically at the end of the day, it's like that Benchling web service API has a good breadth, so we can query, from the samples to the containers and get all that information back and ensure that we get a full picture of what our inventory is and get that an accurate plate definition for them. And again, because of being able to do this all in an automated fashion, this kind of step saves the scientists about 2 days in terms of building those spreadsheets themselves and going into the lab and looking and trying to find those tubes. And the third use case I have is using the Benchling assays, and this is like a thermal shift assay, and so you basically put different conditions and looking at the protein and look at the response, and so they'll take a biorad file and drop it into the assay on the ELN, and then in the back end we'll pull that biorad file, and it's a zip file, it contains multiple files, through a Lambda function, AWS Lambda function, we'll pull that apart, read all the data from that biorad file, import it in as assay results, and then put it, basically it all goes into the data warehouse, and then we have a tie-in to Tableau with the data warehouse, and on the fly we can then generate graphs of all those biorad data, so the scientists can quickly look and manage those assays without having to, previously, they would take those files and they could run locally and manage a file directory structure and keep track of all those experiments. And this basically really centralizes all that. And it also then makes it easier for them to look across, multiple runs and see how their samples or how their assay is working across different conditions. We have some of the infrastructure supports like this more complex methodology where we take a data file and we do like secondary analysis on it, but we also do some very easy what we call simple assays where they just put in some of the data right into their assay, and then we can then just pull that into some Tableau reports or other visualizations to let them see what they're doing. In review, the benchling has really helped us dramatically in three aspects of our goals. In time, with the automation of the sequencing workflow, it really eliminates all the user-driven processes and makes it much more automated to really speed up that turnaround time. And that basically enables us to do more science because we can get more experiments and we can actually, with some of this other automation, not only we've increased the throughput and number of experiments, but the breadth of the experiments as well. It's helped inequality. From the second case study, we're using the Benchling Web Services for designing those primer plates. It really eliminates a lot of that very time-intensive, manually driven processes of going through and looking at your inventory, looking at your primers, and then trying to export data out and putting into a spreadsheet to really understand what you want to pair up for primers to your samples. And so by eliminating the need for a user to kind of go through that by hand, as long as they enter in the data into the system, we can pull that data out and we can generate all those plates and informational messages to them. And finally, for functionality, using the assay module really allows us to capture the data right from the lab and present to the scientists. And using it through visualization tools, it allows us to centralize that data and provide that longitudinal analysis capability. So we can go across from assays and then tie that into samples to all sorts of data, and it doesn't matter whether it's in the benchling warehouse or if we have it supported through AWS Athena. We can basically use the Tableau tool to pull that benchling data warehouse data source in with the Athena data source and blend that together and to create a seamless report across those different silos. I'd like to thank my team. A lot of this was done with a lot of their hard work. And they're here, so you can talk to them, and they could probably give you a lot more details of some of these processes. Thank you.
Hi, Ryan, Bench Therapeutics. I have a question about, you said you used Lambda for some of the dumping of data from Benchling and different sources. What triggers the Lambda job?
We have a variety of different Lambda functions So some of them, like from the sequencing side of things, when the last of the sequencing data files, we have a process that takes it from on-site to push it to S3. And once that last file is sent to S3, that triggers a Lambda function because we have a trigger on S3 to the Lambda function.
Gotcha. So it's a system outside of Benchling itself.
Correct. Yep. But then that triggers the calls to Benchling to generate this analysis sheet because it's something now that we have the data available. I can generate that to push to the next step because I don't want to kick off that analysis until all the data because since we're cloud first, I don't want to push or try to do any analysis to all that data is present in Amazon.
Cool. Thank you.
Hi, Kata Rubius. How many members are on the support team that you have for like database architecture and doing all of this? It seems like you guys are heavily leveraging the API.
Yep. So I currently right now have 3 people. The third just joined me this week, so it had been previously been just me and 2 other people working on the Benchling APIs and all that.
But there was at least always one person that was doing that there?
Yeah.
Okay. All right. Let's try to make the argument that we need that.
It's one of those things. Once you get some momentum, you build it out. Like we're finding that it's easier and easier to build things on top of Benchling and do more of these interesting problems because Benchling handles all the basics for us, and we're left to the interesting problems.
Hi. Question. Other than the Lambda functions, what other tools are you using to connect to Benchling?
So we use the web services, and so a lot of that goes through Lambda functions. We have a few web pages where, like I showed briefly for like the primer plates, and so that basically is just some simple web front end, but that's just calling web services on the back end. So I use Java and Jersey for that to call the web services. And then we also use AWS Batch. And so some of the processing, AWS Lambdas only have a maximum of five minutes. And so like the assay, what I showed, the DSF assay stuff going through the Benchling warehouse, that goes through AWS Batch because when we pull apart that zip file, then process that Bio-Rad data, it takes more than 5 minutes. And so that all just, we basically have a Lambda function kicking off that batch and feeding the assay run information into that, and then it processes it and saves that all into the assay results for us.
Hi, you mentioned in your first slide about instrument integration. Can you explain a bit more on what instrument you integrated?
So we work on we have another tool which I didn't show here, but generating like an echo file for feeding into our echo. We have the ham and the Samilton, the SAM Hamilton freezer integration there. And it's one of those things we're working and building out more, and so we're currently working on getting tying in with some FlowJo stuff, and there's multiple other. Unfortunately, as you probably know, there's no simple API to all the different lab. Each instrument is unhappy in its own way.
Hi, over here. What is your model for training people to use Benchling? Do you do it per group? Is there onboarding when you have new users? How do you handle that?
Yeah, so that's constantly evolving, and so we've had group-based systems where we go through and sit down with users group by group and do training for the ELN. We have a la carte, someone just lets know that they need some help with an assay, and so we'll sit down with that group and do deal with that. We're slowly moving more towards like a weekly kind of open office hours so that people can come, because it's never always easy to know who needs the help. And so if you just make yourself available, hopefully, you make yourself available, they will come. But it's constantly evolving. I don't think there's necessarily a one right answer fits all for that, but yeah.
If Editas is making personalized, maybe personalized genomics medicine, you might be developing like processes that are part of like a therapeutics. And have you encountered challenges translating these from like maybe research processes into GMP processes? Are you able to, I don't know, like if you build something that like enables you to like look at what patient's going on in the patient and then build solutions for them, can that process then be taken out of Benchling into a GMP space?
Well, we're not quite there yet. We're moving towards that right now. We have an IND filing that we're going to do sometime this year, and so some of that will probably we'll figure out as we go. Yeah.